Hello everyone, welcome back to Dr. K. Primer Biotech Lectures. In today's lecture, we'll talk about rDNA technology. As you all know that rDNA technology is all about recombinant DNA technology. Recombinant DNA technology. Today, I'm going to talk about rDNA technology in nutshell. So I may not take more than 10 minutes to explain the what is rDNA technology. So the rDNA technology or recombinant DNA technology has been a buzzword since the discovery of restriction endonuclease and after the production of first recombinant hormone insulin by Genentech in 1981. After that, it became a discipline where it has been used to decipher the structure, function, and regulation interaction of the macromolecules such as genes, proteins, and other macromolecules. Right? So my channel name is Dr. K. PREM-PRIMER Biotech Lectures. Don't forget to subscribe my channel for more updates. And in today's lecture, I will define what is rDNA technology, and I'll try to explain with what is rDNA technology with some examples. You can say it is a technology. Basically, rDNA technology is a technique or a technology used to join the DNA fragments. Here, by using this technique or technology, we are joining the DNA fragments, nothing but genes, which are isolated from different organisms. We are isolating the DNA fragments from different sources and joining them and incorporate into the host. So, in rDNA technology, we will isolate the DNA fragments from different sources and we will join them and incorporate it into the host for a different purposes. Right? We will see that also. Here you see. Why are you incorporating into the host? First thing is that to facilitate the production of more copies of the joined DNA molecule. The purpose of the joining of two different DNA molecules which are isolated from the different sources, when you incorporate them into the host, why are you incorporating? To get the more copies of the same. So that you can use the more copies for different purposes like sequencing or side-direct mutagenesis or preparation of probes or preparation of other molecule. Right? And other way you can say, not only to increase the number of genes you clone, to increase the copy number of the not only to increase the copy number of the cloned gene, but also rDNA technology facilitate to induce the overexpression of genes cloned. When you clone your gene of interest from different source, you want to get a abundant protein, since it is having a therapeutic value or industrial value. So when you identify the value of the gene and its protein in industry or healthcare, then you cannot isolate in a required quantity from the natural sources. So you identify the gene and clone into the suitable vector and transform into the suitable host and induce, so that you get the most high quality and high quantity proteins. So Other purpose of the rDNA technology is to induce the overexpression of genes cloned to produce more amount of protein. This is the second. And you can see third one is that genetic modification. So you can add a new trait to the host. When you identified some gene which is having a value or a very important character, that character you want to bring to the host. In that case, when you clone your gene of that interest into the suitable vector and you transform that vector itself integrated into the host genome, and it will host genome, when it is integrated into the host genome, it can develop a new trait that is called as a genetic modification. You can see integrated into the host. Start. Oh, wait. Third version is that when you clone your gene of interest and you see that there is a value to that gene and you want to add that gene to the particular organism permanently. So ectopic expression and integration expression. So here, in case of production of more copies or to induce the overexpression, most likely they are episomal or ectopic expression. But when it comes to the, you want to add a character to the your desired organism with a desired gene, then you can integrate into the host. You can integrate it into the host genome to develop transgenic plants and animals. You can develop transgenic plants and animals by inserting your gene of interest into the host genome that is permanently, integrated, right? So in the next slide, I'll explain the same thing with a cartoon. Yeah, here you can see this is a plasmid, or you can say vector, and the vector is digested with the restriction endonucleases, and when you digest with the restriction endonucleases, and it get linearized, and you can get a gene of your interest from any source. That's a gene. Then once you have a gene of interest and linearized vector, you can go for ligation with the help of or by employing a DNA ligases. DNA ligases are the enzyme which catalyzes the establishment of a phosphodiesterase bond between 5' phosphates and 3' OH of the two different DNA molecules. Here, when you employ a DNA ligase, and then it can stitch the both linearized vector and gene together to get a recombinant molecule. So such a molecule is transformed into the E. coli. Here you are transforming this molecule into the E. coli, that's a DH5 alpha strain. When you transform into the E. coli, this is an E. coli strain. When you transform into the E. coli, this recombinant molecule will amplify. It become more copies. You can see here. This is your gene of interest. You are inserted into the backbone of the vector, and when you transform to the E. coli, you can see you can get more copies of the same gene. As I told you that by using the recombinant DNA technology, you can increase the number of copies of your gene of interest. That's what is copy number of the clone gene can be increased more than 1,000-fold. This is one application, right? And second application is that I told you that same, if suppose here you want to, this is a suppose insulin gene, right? So you clone into the expression vector, you think it is expression vector. Once you clone into the expression vector, you can transform into, you can transform this construct with the insulin gene into a E. coli or yeast or fungi or insect cell line or plant cell line or animal cell line, right? So you can transform based upon your requirement, right? Based upon the nature of the protein, right? So you can choose the one of the hosts. Then what will happen? You can scale up to the large quantity by using the bioreactor or a fermenter to get a proteins of recombinant proteins. You can see, one is you can increase the copy number of your gene, and second thing is that you can induce the expression of gene to produce a large quantities of the desired gene to into a protein. So you can see recombinant proteins, hormones and interferons, interleukins, growth factors, tumor necrosis factor, blood clotting factors, thrombolytic drugs, and enzymes. So these all are having a therapeutic value. You cannot get them from the natural resources such as humans or animals or any other sources in a abundant. So in that case, you can clone that gene into expression vector and transform into the suitable host and induce that gene expression, get a large quantities of the protein and purify it, and you can use it. This is another application of rDNA technology. The other one, so you want to integrate a gene of interest into the host, right? As you all know that BT cotton that developed by integrating the Bacillus thuringiensis gene, cry gene, into the plant genome. So here they have isolated the gene from the Bacillus thuringiensis and they integrate into the plant vectors and transform into the plant cell. From the plant cell, you can get the whole plant. This plant will express here the gene is integrated into the plant genome and it constitutively expresses that gene, and that gene product is there in the every part of the plant body. Whenever insect eats this plant, any part, organ or part of it, and that will die because the cry protein is going into the midgut of the insect and there it makes the holes, then that insect dies due to the starvation. It cannot take up the food anymore because it is making a holes in the gut. That's an endotoxin. So we can integrate the gene of your interest into your desired host, and you can give a new trait to that particular plant. We never know that cotton plant with a cry protein which can kill the insects, but that is possible with the rDNA technology. And similarly, we can do with the even animals also. So where you can take the gene of your interest, right? And that construct can be injected into the male pronucleus of fertilized oocyte, and that construct undergoes injected into the in vitro cultured early mouse embryo. That embryo is transferred to the mice, and that mice is offspring of mice will be transgenic. So here you can develop a transgenic mice where you can create models for studying of the pathogenicity or to produce the proteins or to get the biopharmaceuticals. Like this, you can add a new character to the animal. They are nothing but transgenic plants and animals. So this is all about rDNA technology in nutshell. So thank you all for listening me. If at all you like it, try to subscribe my channel. That's a Dr. K PREM-PRIMER Biotech Lectures, and try to comment if you have any questions, and thank you all.